Before start this video, please subscribe my channel and click on bell icon. Thanks.
A new study published on June 20, 2023 in PLoS Biology by an international team of scientists including researchers at the University of Bath, finds that our earliest development in the womb may be rather different to what we have always assumed. While human adults are made up of trillions of cells, we all started out as just one cell, the fertilized egg. This divides to become 2 cells which in turn divide to become 4, which become 8 and so on. At some point the cells then start to specialize in their function. Like trains sent to different end stations, some will be shunted off to become the placenta while others will become the embryo. Self-destructing embryonic cell. The team of scientists analyzed previously published data on gene activity of each individual cell from 5-day-old embryos and discovered around a quarter of the cells didn't fit the profile of any of the known cell types, pre-embryo, pre-placenta, etc. Investigating further, they discovered that these cells contain so-called young transposable elements or jumping genes. These are rogue elements of DNA that can copy themselves and insert themselves back into our DNA, often causing damage in the process. Staining of embryos by project collaborators in Spain confirmed the existence of the cells with proteins derived from the jumping genes. Looking a little further forward in time, the team found their descendants both have DNA damage and undergo a process of programmed cell death. Quality control mechanism. This process, the researchers suggest, looks like a form of quality control, selection between cells in favor of the good ones. Dr. Zuzana Isva, K., co-senior author from the Max Delbruck Center and an expert on mobile DNA, said, "Humans, like all organisms, fight a never-ending game of cat and mouse with these harmful jumping genes. While we try and suppress these jumping genes by any means possible, very early in development they are active in some cells, probably because we cannot get our genetic defenses in place fast enough." Co-lead author Professor Lawrence Hurst, from the Milner Center for Evolution at the University of We are used to the idea of natural selection favoring one organism over another. What we are seeing within embryos also looks like survival of the fittest, but this time between almost identical cells. It looks like we've uncovered a novel part of our arsenal against these harmful genetic components. Using old genetic enemies to fight new ones. Conversely, the single-cell data showed that the key cells that will become the embryo, the inner cell mass or ICM, don't contain jumping genes but instead. The authors suggest that if the quality control process is too sensitive, the embryo as a whole may die. This might explain why some mutations in our system to detect damage in early embryos are also associated with infertility. Thanks for watching. Please subscribe my channel for more videos.